Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing DBT JRF Entrance Exam question paper conducted in the year 2023. If you are new to my channel, please do like, share and subscribe. Then next one. Clustering in a gene expression analysis via microarray experiment refers to. So gene clustering, it is an analysis method in DNA microarray data. So what will happen here is clustering algorithms, they will divide the genes into groups according to the degree of their expression similarity. So clustering is cluster of probes used to monitor gene expression. No. Cluster of cDNA is printed on the array. No. Genes clustered together in the genome. No. A set of genes that are likely to work together in a given biological context. That is correct. Answer is option 4. Then next one. Which of the following genetic processes involve sex pili? So it is seen in conjugation in bacteria. Answer is option 1. Then next one. Following are certain statements regarding flowering locus gene in Arabidopsis. Which of the following set of statement is correct? FLC is a strong repressor of flowering. That is correct. Higher expression of FLC induces early flowering. How can that induce early flowering? Because it is a strong repressor of flowering. So B is not correct. FLC is activated by Frigida, FRI. That is correct. Vernalization represses FLC. Vernalization is giving cold treatment. It will repress FLC and it will lead to flowering. So the answer here is A, C and D. Answer is option 4. Then next one. Given below are two statements. Statement 1, statement 2. In the light of the above statement, choose the correct answer from the options given below. So statement 1. Palindromic sequences are DNA sequences that are the same when each strand of DNA is read in the same direction. So first strand it is GAATTC, and in the second strand also from 5' to 3', if it is GAATTC, then it is a palindromic sequence. And palindromic sequence, they are usually recognition sites for restriction enzymes. Then statement 2. Palindromic sequence serve as recognition site for many type 2 restriction endonucleases. That statement is also correct. So both statements 1 and 2 are correct. Answer is option 1. Then next one. Select. Systemic evolution of ligands by exponential enrichment is a useful technique for identifying a ligand-specific oligonucleotide which can be utilized for diagnosis and treatment. The molecule is termed. So Selex, it is a technique for producing oligonucleotides of single-stranded DNA or RNA, and these single-stranded DNA RNA, they specifically bind to a target ligand or ligands. And these single-stranded DNA and RNA, they are called as aptamers. So they have asked here, what are aptamers? So the answer here is option 3, aptamer. Then next one. Which of the following is not true regarding phosphoenol pyruvate carboxykinase type of C4 photosynthesis? Aspartate, this is, see the picture, this is first the mesophyll cell, this is the bundle sheath cell. Okay. Now, aspartate is transported from mesophyll cell to bundle sheath cell. Yes. Aspartate, it is transported from as aspartate to bundle sheath cell where it is converted to PEP, which is returned back to the mesophyll cell. Then alanine is transported from bundle sheath cell to mesophyll cell, Alanine, if you see here, alanine is transported from bundle sheath cell to mesophyll cell. That is also correct. Then oxaloacetate is formed both in mesophyll cell to bundle sheath cell. I don't know what they meant by this statement, but anyway, that is the answer. Oxaloacetate is formed both in mesophyll and bundle sheath cell. Then malate is transported from mesophyll cell to bundle sheath cell. Malate is transported from mesophyll cell to bundle sheath cell. Yes, over here malate, it is transported from mesophyll cell to bundle sheath cell. Malate is converted to pyruvate. Pyruvate is converted to alanine. Alanine back to pyruvate in mesophyll cell. And then PEP formation and the cycle continues. So the answer here is option 3. Then next one. Which one of the following is a neurodegenerative disease? So out of the given option, Huntington's is a neurodegenerative disease. Then next one. Which of the following have been implicated as a potential cause of red wine headaches? So once you consume red wine, some people get headaches. Now why is it so? So enzymatic action in the metabolism of phenols and polyphenols, it will cause fluctuations in the dopamine and serotonin levels, and this will cause headaches. Now dopamine and serotonin, they are called as biogenic amines. So the answer here is option 1, biogenic amine, because it is the fluctuation in these biogenic amines that is causing headache. Then next one. Cholesterol is transported in blood in the form of low density lipoprotein particles that contain apolipoprotein, cholesterol, cholesteroesteryl esters with long fatty acid chain and phospholipids. Where do you think the phospholipids are located in LDL particles? So the question here is, where are phospholipids located in LDL particles? The rest of it is just to confuse you. They have written so many things. Now lipoproteins have a central hydrophobic core of non-polar lipids, that is cholesterol esters and triglycerides. Now this hydrophobic core, it is surrounded by a hydrophilic membrane consisting of phospholipids, free cholesterol and apolipoproteins. So where do you think the phospholipids are located in LDL particle? It is on the surface. It is found outside the hydrophobic core. It is found in the hydrophilic membrane outside the hydrophobic core. So the answer here is option 2. Then next one. CPG islands are sequences present in eukaryotic DNA. This is statement 1. Then statement 2. Promoter regions of the many expressed regions in eukaryotes are generally enriched in CPG islands. Choose the correct answer from the option. Actually, both these statements are correct. So the answer here is both statement 1 and 2 are true. Then next one. Polyhydroxyalkanoates produced by microorganisms have application as bioplastic. Answer is option 2. Next one. Which of the following structures is not seen in cells of angiosperms? So angiosperms are plant cells. They have particularly asked angiosperm just to confuse you all, because the answer is centrioles. Okay, centrioles are not seen in the cells of angiosperm. They are found in animal cells and lower plants, not in higher plants. Higher plant is angiosperm. Lower plant is your chlamydomonas. So in chlamydomonas and in animal cells, these centrioles are present, but they are not present in angiosperm. So instead of asking plant cells, they have asked specifically angiosperms just to confuse you all. So the answer here is option 1, centrioles. Then next one, CRISPR-Cas9 technology is typically used for. So it is typically used for gene editing. Answer is option 2. Now next question. Match photosynthetic proteins in list 1 with their respective encoding genes in list 2. So A will be PSBA, D2 will be PSBD, CP47 will be PSBB, CP43 will be PSBC. So the answer here is A4, B2, C1, D3. Answer is option 2. Then next question. The annotation of a gene sequence is generally stored in which of the following file formats? So it is GFF format it is stored, that is general feature format. Answer is option 2. Then next one. Which of the following is not a type of G protein coupled receptor? So G proteins, they are classified into 4 families according to their alpha subunit, that is GI, GS, G12 or 13, G and GQ. The GS and GI family regulate adenyl cyclase activity. GQ activates phospholipase C beta. G12-13 activates small GTPase families. So they have asked which is not a type of G protein. So GP is not a type of G protein. GQ, GI, GS are types of G protein. So the answer is option 4. Then next one. Signal propagation at a chemical synapse is characterized by. So they have asked chemical synapse. So it is unidirectional and there is delay also. So delay and unidirectional signal propagation, that is correct, whereas A is not correct. They have given bidirectional signal propagation. It is not bidirectional signal propagation. Then C, efflux of Ca2 plus ions leading to release of neurotransmitter at the presynaptic terminal. It is not efflux of Ca2 positive ion, it is influx of Ca2 positive ions. Then next one, significant influx of Ca2 positive ions at the postsynaptic neuron. It is not postsynaptic neuron, it is presynaptic neuron. So D is also not correct. So C is not correct. D is also not correct. So the answer here is B only, option 2. Then next one. Which of the following can be used for the discovery of unknown conserved motifs in a given set of DNA or protein sequences? So there are discovery of unknown conserved motifs. So BLAST, it finds regions of local similarity between 2 sequences. Then Philip, it is for phylogenetic analysis. Clustal, it is for multiple sequence alignment. So what remains? ME, option 4. So the answer here is option 4. ME. Next question. Which one of the following does not directly account for water potential in plants? Pressure, yes, it accounts. Temperature, it doesn't directly account for water potential in plants. Osmotic components and gravitation. So answer here is option 2, that is temperature. Then next one. Given below are 2 statements. One is labeled assertion A, other is labeled as reason R. Assertion A base substitution in DNA does not always result in mutant phenotype. Yes, because, okay, we'll see the reason. Some base substitutions in codons do not change the encoded amino acid. Okay. So base substitution in DNA does not always result in mutant phenotype is correct, and R is the correct explanation of that assertion. So in the light of the above statement, choose the most appropriate answer. So both A and R are correct, and R is the correct explanation of A. Then next one. Which one of the following laboratory tests would be the best to determine the number of CD4 positive cells in the blood of patient infected with HIV? So this is a direct question, so it is flow cytometry. Then next one. Match the plant source with the corresponding secondary metabolite. Belladonna. Belladonna is atropine. A2. A2 is found in option 1 and option 3. Foxglove. Foxglove is digitalin. Pacific U, that is Taxol, and eucalyptus is menthol. D1 and C4. So the answer here is option 1. Again match the list 1 with list 2. That is next question. List 1 they have given bacterial products and list 2 they are popular application. Nicene, it is a bacteriocin, so it is naturally involved in preservation of various foods. A is option 2. 1 and 4 you have to see. The 2 and 3 is not correct. Superoxide dismutase. Superoxide dismutase, it is an antioxidant. B1. So option 1 itself is correct. Then polysaccharides. C4. Gums and thickeners, culture viscosity stabilizers, and probiotic cultures, lactose digestion, control of intestinal pathogens. So the answer here is option 1. Then next one. Which one of the following is not considered as a secondary messenger? CAMP is a secondary messenger. ATP is not a secondary messenger. Ca2 plus and IP3 are also secondary messengers, whereas ATP is not a secondary messenger. This year's DBT question paper was comparatively easy when compared to that of the other years. Most of the questions were direct questions. Okay, next one. Equine embryo collection is commonly performed on day 7 or 8. Day 0 equal ovulation because recovery rates are high and the blastocyst can be easily recovered and handled. Answer is option 3. Then next one. Biological oxygen demand, BOD, refers to the amount of oxygen that bacteria consume during decomposition of organic matter in aerobic conditions. Answer is option 3. Then which of the following blast search program is used to find the putative function of a given nucleotide sequence to search a protein sequence database? So here it is nucleotide sequence is the query and protein sequence database is the where you have to compare this particular nucleotide sequence with. So it is BLAST X. First option itself is correct. BLAST P is protein sequence is the query and protein sequence is the database. BLAST N is nucleotide sequence is the query against nucleotide sequence in the database. And BLAST Z, I didn't find anything like BLAST Z. I don't know whether it is there, but I didn't find anything. So BLAST X is the answer. Then next one. Enzymatic production of aspartame, a low calorie sweetener, involves the use of. So it involves the use of thermolysine. Answer is option 4. Then next one. During genome assembly, which of the following sequence is generally followed to obtain good chromosome level assembly? So genome assembly, it is the process of taking large number of short DNA segments or DNA sequences. Then you put them back together to create an original chromosome, to create a representation of the original chromosome from which you took the DNA sequences. So from DNA you are going back to the chromosome. That is called as genome assembly. So what are the sequences will be? So it will be reads, contigs, scaffolds and then chromosome. So the answer here is option 1. Then next one, list 1 match list 1 with list 2. List 1 is protein and list 2 is their function. So DNA A. DNA A, it recognizes ORI sequence, opens duplex at specific sites in the origin. So DNA A is option 1. DNA B. DNA B unwinds DNA. DNA G synthesizes RNA primers and DNA gyrase. It relieves torsional strain generated by DNA unwinding. So the answer here is A1, B2, C3, D4. Answer is option 1. Then next one. Which of the following is not true for microalgae? So microalgae can perform photosynthesis only if the light source is available. That is correct. Respiration by microalgae continues throughout light and dark period. That's also correct. Photosynthesis by microalgae is an oxidation reaction. Photosynthesis is a light-driven redox reaction, and in here carbon dioxide is converted to carbohydrates and oxygen is released. So it is a redox reaction, not an oxidation reaction. So which is not true? Option 3 is not correct. Then next one. The intrinsic apoptotic pathway is primarily regulated by. So the intrinsic apoptotic pathway, it is regulated by BCL2 family proteins, and BCL2 it is found on the outer membrane of mitochondria. So the answer here will be option 3, that is mitochondria, because BCL2 is found in mitochondria. Then what are proto-oncogenes? So proto-oncogenes are oncogenes which can become cancerous, or they can become oncogenes. Answer is option 3. They can become oncogenes. So the answer is option 3. Then next one. Many bacteria produce an extracellular capsule composed of a network of polysaccharides and or proteins that are loosely attached to the cell wall. Yes, you can find many bacteria with capsules. And these capsules, they are loosely attached to the cell wall. Which of the following statement is incorrect about the capsule? It is a virulence factor. Yes, if you remember Griffith experiment, you come to know that it is a virulence factor. Many potentially inhibiting, but not a transformation factor. Okay, transformation factor in Griffith experiment was DNA. Many may potentially inhibit binding of host complement proteins. That's also correct. Helps the bacteria to evade the host immune system. That is also correct. It prevents the pathogen from acquiring nutrients. It doesn't prevent the pathogen or bacteria from acquiring nutrients. So the answer here, which is incorrect, they have asked, it is option 4. Then next one. Following are certain statements regarding gibberellin signal transduction in Arabidopsis plant. So they have given DELA, GA, G1, D1. Okay, we'll see what is all these things. Now first thing is DELA is a repressor of plant development. Now how do you repress the activity of this DELA? How does gibberellin repress the activity of this DELA? So when gibberellin, it binds to G1D1 receptor. So G1D1 is the receptor for gibberellin. It enhances the now this binding, it enhances the interaction between G1D1 and DELA proteins. Now what will happen once this G1D1 DELA complex forms? Now what will happen is, once this complex is formed, it promotes the ubiquitination of DELA proteins and thereby preventing their degradation by the 26S proteasome. So once DELA is degraded, the cells get released from their repressive effect, because DELA is a repressor of plant development. Now we will see what are the statements. DELA is a negative regulator of growth. That is correct. GA is not required for interaction of delta with G1 D1. It is required. Only after this GA G1 D1 delta complex forms, delta will be ubiquitinated and degraded. So B is not correct. G1D is a receptor of GA. That is also correct. Then delta is ubiquitinated only after GDI1 perceives GA and binds to delta. That is also correct. Proteasomal degradation of delta is required for the growth of plant. That is also correct. So A, C, D, E are correct. Answer is option 3. Then next one. The antigen binding site on antibody is formed primarily by. So this is the antigen binding region. So it is having hypervariable regions of both heavy chain. This is your heavy chain and this is your light chain. So the hypervariable region of both heavy and light chains, they contribute to antigen binding site on an antibody. So the answer here is option 1. The next one. AlphaFold is an artificial intelligence based method for. So it is a method to perform protein folding based on a model trained on existing protein structures. Answer is option 3. Then next one. Which of the following processes turn on and turn off the alpha subunit of a heterotrimeric G protein? So this is the cycle of a heterotrimeric G protein. This is the receptor. This is where the ligand will bind. In the beginning stage, how it will be is that is under basal condition, G protein exists in the cell membrane as heterotrimers composed of alpha, beta, gamma subunits. They will not be bound to the receptor. Alpha subunit will be bound to GDP. Now once the receptor is bound by the ligand, the trimeric protein gets attached to the receptor. And the GDP will be replaced by GTP in the alpha subunit. Once it gets bound to GTP, alpha subunit detaches from the receptor and it will also dissociate from the gamma and beta subunits. Now this alpha subunit bound to GTP and the gamma and beta subunit, they both regulate certain effector proteins like ion channels, adenyl cyclase, phospholipase C, phospholipase A2, phosphodiesterases. Once the ligand is being removed from the receptor, the GTPase activity that is present in the alpha subunit hydrolyzes GTP to GDP. Once alpha become bound to GDP, alpha, gamma and beta subunits will join it and they will remain as heterotrimeric form like this in the how it was in the beginning. So that is the cycle. Now coming to the question. Which of the following processes turn on and turn off the alpha subunit of a heterotrimeric G protein? So binding of GDP. Binding of GDP, it was in the basal state. Alpha subunit in the basal state. So there wasn't any turning on of the alpha subunit. Now when did alpha subunit turn on? It's turned on once it got bound to GTP. So binding to GTP, that is correct. Exchange of bound GTP with GDP, it was turned off. It was not exchanged, it was GTPase activity of the alpha subunit which hydrolyzed the GTP to GDP. So third option, if you see binding to GTP, hydrolysis of bound GTP. That is correct. So the answer here is option. And next one, Recce protein is involved in. So it is involved in recombination. Answer is option 2. Then a Phred score is of 30 in a DNA sequencing output refers to the probability of incorrect base call as 1 by 1000, 1 in 1000. So Phred quality score is used to assess the accuracy of sequencing platforms. And it indicates the probability that a given base is called incorrectly by the sequencer. So if it is 30 means it will be 1 by 1000. If it is 20 means, if the Phred score is 20 means it will be 1 by 100. Now 1 by 1000, this means that the base call accuracy, that is the probability of a correct base call, is 99.9%. So the answer here is 1 in 1000. Then next one. Which of the following statement is most appropriate with regards to the primary and secondary antibody responses? The IgM made in the primary response is made primarily by memory B cells. Not by memory B cells, it is by plasma cells. Then next one. So this statement is not correct. The lag phase is shorter in primary response than in secondary response. Secondary exposure causes memory cells to form and not primary response. So, secondary exposure occurs because of the presence of memory cells. Okay, so secondary response will have a shorter lag phase, whereas primary response, it is having a longer lag phase. So option 2 is also not correct. In the primary response, memory B cells are produced but memory T cells are not. In primary response, both memory T cells and B cells are produced. Then next one. The amount of IgG made in the secondary response is greater than the amount made in the primary response. That is correct. So the answer here is option 4. Then next one. Which one of the following cells is involved in translocation of microRNA in plants? It is phloem sieve elements. Answer is option 3. Then next one. Which of the following pigments makes some archaea to appear purple? It is bacteriorhodopsin. Answer is option 4. Then next one. Which of the following is a nucleic acid hybridization based test for diagnosis of tuberculosis? So it is TrueNAT. First option itself is correct. TrueNAT test is a chip-based real-time PCR test for semi-quantitative detection and diagnosis of Mycobacterium tuberculosis obtained from the sputum samples of human. That is TrueNAT test. Then TORCH screen, it is a group of blood test and it is done usually in neonates, from pregnant women or children who are born in and who are 1 or 2 days old. They are neonates. So the TORCH, full form is toxoplasmosis, rubella, cytomegalovirus, herpes simplex virus and HIV. And Vidal test is for typhoid fever, and Mantox tuberculin test. It is a method for determining whether a patient is infected with Mycobacterium tuberculosis. So the answer here is option 1, TrueNAT. Then next one. An anti-malarial drug such as primaquine is believed to act by causing oxidative stress to the parasite. Okay. This drug is not suitable for treatment in individuals having deficiency in. So if the individual is deficient in glucose 6-phosphate dehydrogenase, they won't be given primaquine treatment. Why? Because It causes severe hemolysis in patients with glucose 6 phosphate dehydrogenase deficiency. So primaquine is not given to such patients.